Hello everyone. Assalamualaikum warahmatullahi wabarakatuh. Welcome back to Food Technology Study Program, Department of Food Science and Technology, Faculty of Agricultural Engineering and Technology, IPB University. My name is Rati Dewanti Haryadi. I am a professor in food microbiology, and today we will meet again to discuss our lecture ITP 322, Food Safety and Sanitation. This week, we are going to discuss our 10th topic on process control that include validation, verification, and environmental monitoring. We will be discussing process control in food industry, the difference between validation, verification, and monitoring, tools to assure appropriate control measures and food process control, as well as microbiological criteria. Upon completion of this lecture, you are expected to be able to define process control in food industry, differentiate validation, verification, and monitoring, describe ways to assure control measures and process controls, explain several control measures that have been successful in controlling foodborne pathogen, and describe environmental monitoring program. Let's start with process control. Process control is assuring that all processes, especially those related to the kill steps of microorganisms, are properly carried out to achieve the objectives. For example, if we are going to make a pasteurized milk, we want to be sure that the pasteurization steps can kill, for example, 5 log of microorganisms. Generally, process control involves validation, verification, and monitoring. So what is the difference between validation, monitoring, and verification? Monitoring is defined as the act of conducting a planned sequence of observation or measurement of control parameters to assess whether a control measure is under control. Meanwhile, validation is obtaining evidence that a control measure or combination of control measures, if properly implemented, is capable of controlling the hazard to a specified outcome. Verification is the application of methods, procedures, tests, or other approaches in addition to monitoring to determine whether a control measures have been operating as intended. So for example, if we are going to pasteurize milk, during the pasteurization, we are going to have somebody monitor whether, for example, a temperature of 80 degrees for 15 minutes is achieved. Validation means we are doing experiments, for example, or predictive microbiology to evaluate whether 80 degrees Celsius for 15 minutes could kill, for example, 5 log of targeted microorganisms, say Salmonella. And verification is when we already operating the process and take our samples and check whether Salmonella is not present in our products. These are several means to assure control measures and process control. Validation of control measures, verification of process control, verification of environmental control, corrective actions to re-establish control, microbiological testing in customer-supply relation, and end product testing. So what do we do during the validation of control measures? These are some of the activity: collecting results of evaluation, collecting scientific and technical information, observation and testing, including control measures applied at supplier and by the consumers. All control measures to control the initial hazard, H0, reduction of hazard, sigma R, increase in hazard, sigma I, has to be validated to ensure they deliver objectives. Validation can be carried out by predictive modeling, microbial challenge trials, and application of process criteria. Here are the activity during assessment for initial number of microorganisms. Questions that may arise include: is hazard pathogen present in the main or minor ingredient? Is there any seasonal effect? For example, there's a different load of microorganism during winter season versus dry or summer season. Is there any influence from the geographical source? When hazard is unavoidable, then we have to establish a microbiological criteria. Information to validity of incoming materials could include baseline data from government, document from supplier, baseline data of the food processor, or compilation of the testing results. Validation of inactivation studies can be done by predictive microbiology using a model to predict growth, survival, death as a function of temperature, pH, water activity, etc. There are also software available such as USDA Pathogen Modeling Program, Combase, etc. We also can do the validation using a microbial challenge study, generally by choosing a pathogen of target or its surrogate consisting of 5 strains, inoculate with a high number of bacteria, million to 10 million colony forming unit per gram, and the study has to be done to cover the shelf life periods with a minimum of 5 to 7 data points and 3 replicates. Analysis of samples and data interpretation then is done. Challenge studies can also be done to validate the growth of the microorganism of concern. Generally, the study is done by low inoculum concentration, such as 100 to 1000 CFU per gram, in a product formulated and stored similar to the real conditions by considering the lag phase, exponential stage, and consider the competition that may occur by other microorganisms. Food products undergoing kill step may also get contaminated afterwards. However, there are not many references or models to validate or to predict this recontamination. Therefore, validation of the recontamination studies is done by obtaining samples after kill step to assess possible recontamination, also by studying the environmental condition, as well as assessment of the packaging material. These tables show you several control measures that have successfully controlled several foodborne pathogens, such as Bacillus cereus, Brucella, Campylobacter, Corynebacter, Clostridium botulinum, as well as Clostridium perfringens. There is also similar table suggesting what bacteria that can be controlled and what type of control measures that have successfully been acknowledged to control the pathogen. In this table, you can see several control measures that have successfully controlled Vibrio cholera, Vibrio parahemolyticus, Vibrio vulnificus, and Yersinia enterocolitica, as well as viruses such as hepatitis and norovirus. The following list are control measures for parasites such as Trichinella, Toxoplasma, as well as toxigenic mold such as Aspergillus, Fusarium, and Penicillium, and also biotoxins from seafood. In explaining the difference between validation, verification, and monitoring, I use pasteurization as an example in which the parameters for the control are usually temperature and time. In this table, we are learning that a different process parameter has to be applied for different technologies. And here are several of the emerging technologies. In food service industries, the parameters of the process control is probably simpler. You can see there are pressure involved and temperature, and for the others are mostly temperature and time. So how do we know that the validated process actually deliver or achieve the objective? This activity is called verification, and the main activity during verification is testing. Usually, we establish a microbiological criterion to describe what objective we would like to achieve for the process. To do that, we need some information, for example, the level or frequency of the target microorganisms, variability of microorganism within lot and between lot. And we established a control chart per period, for example, yearly or 6 monthly, etc. The followings are an examples on how you can use your data from your testing for verification to evaluate the quality of your lot. The first graph shows you three different lots. The red color represents a very poor lot with a very high variability of the quality, while the green curve shows a good lot with a low or a very narrow standard deviation. You can also use your daily or weekly testing data to evaluate the quality of each lot and see how many times it exceeds the expected values, for example. Sometimes you got recontamination in your product after the kill steps because of the addition of ingredient or because of contamination from the environment. So one other means to assure the process control is by monitoring the environment. Maintenance of a hygienic processing environment is essential for the production of safe and wholesome foods. The principles in environmental control include preventing pathogens from entering the facilities. If they enter, prevent them from establishing themselves. If they manage to establish themselves, prevent them from growth. But if pathogens are persistent in food facility, so corrective actions has to be done. Environmental monitoring can be done by monitoring the food contact and non-food contact surfaces to verify that environment is under appropriate hygienic control. You can use the swab, for example, or sponge or ATP-based assay to verify your environment. Additionally, you can also monitor the air to assure that air involved in processing is not a source of recontamination. Testing of air can be done by collecting air samples by sedimentation method, impaction, filtration, or centrifugation, followed by plating on agar. To verify that environment is under control, food producers or industry usually develop an environmental monitoring program or EMP. EMP is developed based on the pathogen of concern or relevant microorganism, either is the surrogate or indicators. It also has to review all available steps to prevent entry of pathogens, all hygiene control programs, as well as all historical data, including conducting investigative sampling when necessary. EMP also established sampling methods, sampling points, and their microbiological criteria. EMP also established testing frequency and methods, as well as corrective action and periodical review of the data. Another means to assure process control and control measures is by establishing corrective actions to re-establish control. These are done by assessing control of GMP and HACCP plan, corrective actions for GMP, SSOP, and HACCP plan, responding to epidemiologic evidence and complaints, option for disposition of questionable products, for example, by conducting sublot testing, and addressing repetitive loss of control. The other tools is microbiological testing in customer-supplier relation. A good food manufacturer must consider the raw material used. They have to know that raw agricultural commodity, for example, generally has higher variability in terms of quality or, for example, the number of microorganisms in the commodity. Meanwhile, processed ingredients generally has a less diverse or variability in terms of quality. Good company also have a good interaction with the retailers. They may establish contract manufacturers. They perform auditing for the suppliers and microbiological testing, especially when the knowledge about the quality of the raw material is inadequate. The last resort to assure control measures and process control is end product testing. It is acknowledged that end product testing consists of some limitations, such as sampling. We never test all of the product. The late result, we will not know what happened after a few days. And positive results mean contamination present in the lot, but negative result does not mean that contaminant is absent in the lot. And product testing is useful to verify whether food safety control deliver the purpose. For all of this testing, we need a microbiological criterion or microbiological criteria. So that's what we are going to discuss after this. Microbiological criteria, by Codex definition, is a criterion that defines acceptability of a product or a lot based on the absence or presence or the number of microorganisms, including parasites and/or metabolites per unit, volume, area, or lot. A microbiological criterion usually have these components. First is N, which is the number of unit samples to be analyzed which are taken randomly and independently. C is the maximum number of unit samples allowed to contain unsatisfactory results, for example, presence of a certain microorganism or number exceeding little m. Little m is a microbiological limit in a two-class sampling plan that separates good products from poor or bad products. Or in a three-class sampling plan, it separates good products from the marginal ones. Meanwhile, big M is a microbiological limit in a three-class sampling plan that separates marginal product from the poor products. Value equal to or more than big M is not acceptable. Here is the graph of two-class sampling plan, and the microbiological limit here is a little m that separates good product from the poor product. Sometimes a lot can be divided into three groups, that is called three-class sampling plan, that has two limits, that is the little m that separate good proportion of the lot with the marginal proportion of the lot, and large M or big M that separate marginal proportion of the lot with the bad or poor proportion of the lot. Here are an example of microbial criteria in a customer-supplier relationship. For example, for a spices as a raw material, an industry requires the following: Salmonella with an N of 5, C of 0, and little m negative in 25 gram. This criteria means that 5 samples are taken randomly and independently. Each is analyzed for the presence or absence of Salmonella in 25 gram of samples, and all 5 samples have to yield a negative result for Salmonella. And here is an example of microbiological criterion in environmental monitoring. For processing environment of a powder infant formula factory, for example, the guidance in a company is as follows. For Enterobacteriaceae, N is 10, C is 2, and little m is negative per centimeter square. That means that 10 area samples are swabbed according to the EMP. Each is analyzed for the absence or presence of Enterobacteriaceae per 1 centimeter square. 8 samples have to yield negative results per 1 centimeter square, and a maximum of 2 samples are allowed to have a positive result. The following is an example of microbiological criteria applicable in end product testing. According to the Peraturan Badan POM 13/2019, for pasteurized milk, the microbiological criterion for total pet count is n equal to 5, C equal to 2, little m 10 to the 4th or 10,000 per mil, while big M is 10 to the 5th or 100,000 per mil. That means 5 samples are taken randomly and independently. Each is analyzed for total plate count per mil. 3 samples have to have TPC of less or equal to 10 to the 4th per mil, while maximum 2 samples are allowed to have total plate count between more than 10,000, but it has to be fewer than 100,000 per mil. And this is the last example for microbiological criteria in end product testing according to Codex 2008 for powder infant formula. It requires Salmonella with an N of 60, C of 0, and little m negative in 25 grams, while for Chronobacter spp, N is 30, C is 0, and little m is negative per 10 grams. So the standard for Salmonella means that you have to take 60 samples taken randomly and independently, and each is analyzed for Salmonella in 25 grams of samples, and all 60 samples have to yield negative results. Now, can you tell me what the standard for Corobacter spp means? That's all for today's lecture on process control. I hope you understand more about how to make sure that your control measures and your process control can deliver the product that you want. Thank you for your attention. Assalamualaikum warahmatullahi wabarakatuh.